respected chairman of the session, and all distinguished teachers. Good morning to all of you. Myself, Narendra S Salunkhe, PhD student under the supervision of Dr. S.B. Patil at School of Life Sciences. Today, I present my PhD, part of my PhD work entitled Membrane-Bound Yellow Xanthomannate-Mediated Synthesis of Gold Nanoparticles and its Application. Xanthomonas infects citrus family plants. This is a worldwide area citrus plants in grown. 80% of citrus plants get affected by Xanthomonas species. This is a worldwide area. This explore diversity of Xanthomonas and its potential of its pigment. The research data showed that there are hardly very few research papers available on Xanthomonas species for xanthomannate pigment production. Xanthomannate pigments are as multiple functions, including protection against oxidative stresses, UV lights, and other environmental stresses. On the basis, I set up my objectives. First of all, isolation and screening of xanthomonas species for xanthomannate pigment production, then characterization of produced pigments, then effect of different media component on pigment productions, then explore its application in different fields. To achieve all design objectives, this methodology is used. Isolation of microbes for xanthomannate pigment productions. On the basis of yellow color intensity, I have screened xanthomannate pigment producers. Then extraction and purification of xanthomannate pigment on the basis of polychromatography. Then optimization of physicochemical parameters by using 1 factorial methods like pH, temperature, carbon source, nitrogen source, growth mediums we are optimized for xanthomannate pigment production. Then the purified pigments were characterized by analytical techniques and export its application in different fields. First of all, I have collected infected plant material and proceed to routine microbiological technique like preparation of suspension. Stacking was done on Xanthomonas isolation car. On the basis of yellow color intensity, I have screened xanthomannate pigment producers. The selected isolates were used for solvent extraction. Among the ether show the maximum xanthomannate pigment production and best solvents for xanthomannate pigment extractions. These solvents were optimized and used for further experimental purpose. On the basis of yellow color intensity and absorption of maxima, Xanthomonas SMP1 shows the maximum absorption of maxima. On the basis, these Xanthomonas species were used for further selected and used for further experimental work. Then, the extracted pigment was characterized by analytical techniques like spectroscopy, thin layer chromatography, FTIR, NMR, and HRNC-MS. In the UV spectrophotometry was done, the absorption maxima at 450 nanometer and RF value 0.7 was determined. The compared to absorption maxima and RF value of pigment similar to previous reports. Then in the FTIR, picture represent the functional groups of pigment like ketone and hydroxyl groups were there optimized, and it's similar to previously reports. After the FTIR analysis, after the FTIR confirmed the functional groups of pigment, proceed to next characterization like H1 NMR, and it determined the chemical and it determined the total hydrogen atoms, the total 28 hydrogen atoms was determined and support chemical structure of xanthomannate pigment. In the mass pigment, the 1034 Dalton was analyzed by using HRLC-MS. After the characterization of xanthomannate pigment, proceed to the next characterization, one more application like the optimization of xanthomannate detection medium by using physicochemical parameters. The physicochemical parameters by using like growth medium, incubation time, effect of temperature, effect of pH, effect of carbon source, effect of nitrogen source, and their concentration optimize the production medium for xanthomonadic production. The production medium, design a production medium, and the that is peptone 20 gram per liter, glucose 10 gram per liter, pH 1, and temperature 28 degrees Celsius, and incubation time 48 hours were optimized, and 24.46 milligram xanthonidin pigments were produced. After the production of xanthonidin pigment we are used for the application. We are explore its application in different fields. First of all, the pigment was tested in toxicity testing. Pigment toxicity testing against human bloods and aquatic animal like Daphnia magna species. They didn't show any toxic effect of xanthonidin pigment on the basis of the research. They didn't show any toxic effect against Daphnia magna species and human blood species. Hence it used for the further experimental. It's suitable for the further experimental work. Then this is a one more application of xanthonidin pigment, like synthesis of gold nanoparticles. It's a rapid synthesis method. The gold fluoride solution react with the xanthonidin pigment with the presence of sunlight. The nanomaterial was prepared. The prepared nanomaterial were characterized by UV spectrophotometrically, FTIR, particle size analyzer, and electron microscopically. First of all, figure A represents the comparative spectrum of pigment-based nanomaterial and gold solution. Then figure B represents the optimized concentration of dispersed and stable nanomaterial portion. That is 1 millimole concentration was used for the dispersed and stable nanomaterial. Then figure C represents the incubation time optimized for dispersed and stable nanomaterial incubation, that is 25-minute incubation time we have optimized for dispersed and stable nanomaterial incubation. Then the effect of concentration of xanthonidin pigment, the concentration of xanthonidin pigment, required concentration of xanthonidin pigment, we are optimized for the dispersed and stable nanomaterial preparations, that is 15 microliters concentrations we are used for the microgram concentration we are used for the dispersed and stable nanomaterial preparations. After the UV space characterization, proceed to next characterization like electron microscopically, and that is the shape of nanomaterial was determined, spherical shape was determined, and the Eden's graph represents the gold elemental peaks. After the electron microscopic, proceed to next characterization like particle size analysis, and the particle size was determined by 68.9 nanometer, and zeta potential is a minus 12 millivolt was determined. After the zeta potential particle size analyzer, one more next characterization like FTIR analysis, and the FTIR, it's supposed to determine the its possible mechanism of pigment react with the gold solution. Pigment present also negatively presence of the ketone and hydroxyl groups, and these groups interact with gold fluoride solutions, and chloride solution into bulk gold chloride solution into nano gold formation was created, and nanomaterial was prepared. The prepared nanomaterial stabilized nanomaterial was determined. The prepared nanomaterial explore its application in different fields like SPF enhancer, that is SPF enhancer. By using pigment uses as sunscreen protection factors by using Mansour et al. Methods, and the two sunscreen formulated sunscreen 1 and sunscreen 2 formulation, and enhance the effect of SPFs like 3 to 4 degrees as compared to the pigment based sunscreen and standard sunscreen. After the SPF enhancer, I have proceeded to one more application like antioxidant potential. Antioxidant potential pigment have to antioxidant potential. It's a different antioxidant activities like different scavenging activity like DPPS scavenging activity, phospholmolybdenum scavenging activity, and hydrogen peroxide scavenging activity as compared to the standard antioxidative agent, and it was shown the significant antioxidant activity was determined. After the antioxidant potential pigment have to one more application like antimicrobial activity, like antibacterial activity, different gram-positive and gram-negative bacteria we are tested like Bacillus subtilis, Escherichia coli, Pseudomonas aeruginosa, and Staphylococcus aureus we are tested, and it was observed that gold nanomaterial have to deter any type of antibacterial, antimicrobial activity was not determined. After the antibacterial activity, I have proceeded to pigment and pigment-based nanomaterial have to test it in vitro testing for anti-cancer activity against the different cell lines like MCF7, HeLa cells, and SIHA cells. And it was observed that it is a significant for it was significant for anti-cancer activity against MCF cells. This figure represents and support to the tabular data. Conclusion, potent pigment producer Xanthomana species was isolated and identified by Xanthomana's capacitor species by morphological, biochemical, and molecular analysis. Then physicochemical parameters was high pigment production were optimized, that is peptone glucose medium at pH 7 and temperature 28 degrees Celsius and incubation time was 48 hours. Then the pigment was extracted by using column chromatography. Then the pigment characterized by visible spectrophotometer, FTIR, HRNCMS, NMR, TLC. Then the purified pigment was non-halolytic, non-toxic, and had antioxidant, antimicrobial, SPF enhancing potential. Then XP-mediated gold nanomaterial who has enhanced antibacterial activity, anti-cancer potential, and compare with xanthomanatic. These are the references and these are the references. Thank you.